is go over the extra credit provision, which I sent you in a post on Canvas. To do that, I need to share my screen. And now you should be able to see this document. Can you see student participation, extra credit?
Yes.
Yes.
See that? Okay. I would just like to review this with you and tell you why. We're obligated, we have no choice but to be online. But learning online is difficult, as is teaching online. So I'd like to do whatever I can to facilitate learning. And one possibility is to promote class participation. I'd like to have you speak up, whatever's on your mind, ask questions, answer questions, make comments if you have them, complain about problems if you perceive them, point out flaws, whatever. And hopefully learning will be better if we interact verbally. To promote that, I will award 3 extra credit points if you participate at least once in any particular class. So like this evening, if you speak up once or more times, you'll get 3 extra credit points. Now to do that, this is a little awkward, but I will ask you to give me your last name before you pose your question so that I can make sure that I get it recorded so that I can award you the extra credit points. Now, for each student, there are 2 class periods a week. So that's a possibility of 6 extra credit points per week. Over the course of 15 weeks is a maximum of 90 extra credit points for the entire semester. And that will be about 10% of your total grade. Not insignificant. It could be the difference of as much as a grade point in the end. So it's an inducement to participate. And what I would like is for each student in the class to be involved in every class. So some students are more inclined to speak up than others, but even those of you who are shy, I strongly encourage you to participate. Any questions about that right now?
No.
Everybody understands? Let's see. I'm admitting more people from the waiting room. All right, then I'm going to stop sharing that. There is a quiz, an ungraded quiz is available all day today. Has anybody taken it?
Yes.
Yes, I did.
Anybody have any problems with it?
Nope.
I do. I did not have time to complete my last question, which is the writing one.
Okay. What's your name, your last name, please?
YUN.
Okay, you didn't have enough time.
Yes.
Okay, anybody else didn't have enough time? Okay, it's good to know because I have to try to adjust the questions and the type of questions to the amount of time that is allotted. Okay, the other thing I want to do today is go over the worksheet the methods of scientific inquiry worksheet, but I'd like to do that at the end if there's time. But before doing that, I'd like to go through a slide set on methods of scientific inquiry. And to do that, I need to,
Professor,
yes.
my name is Samira Al-Muhammad. We
Good.
have enough time. It's 2:12 midnight, yes, for quiz.
What's your last name again?
Al-Muhammad.
Muhammad?
Al-Muhammad.
Al-Muhammad.
Yes.
Okay, I've got you. And where are you?
It's enough time. It's the 2:12 at midnight, yes?
Okay, it's midnight. All right. And are you having problem
Oh, no,
accessing
just
the
I want
quiz?
to make sure. No, just I want to make sure. It's all the Wednesday, anytime, 10
Yes.
minutes.
Yes, it's
Okay.
all, you'll have until midnight Pacific Coast time
Mhm.
to take this quiz. You want to log in before midnight because at midnight is when it
Yes.
closes.
Yes. Okay, yes, I know that, but I want to make sure it's all the day to midnight. We have enough time. Okay.
Yeah, that's correct.
Thank you. Thank you.
Okay, anything else?
Yeah. My name is Michael Zaror. I was wondering, are all like the worksheets going to be due before classes Wednesdays at 4 o'clock? Is that going to be standard or will it change?
I don't know that that will be standard. I didn't expect anyone to actually fill out the worksheet and return it to me because it's not graded and we'll use it as a basis for a class discussion. But in the future, if I issue a worksheet that is graded, then yes, you will have to return it to me by the closing date or closing time. And that could be just before class, most likely just before class, because I'd like to discuss worksheets in class.
Got it. Thank you.
Okay.
Yes, I had a question.
Okay, and your name?
Sierra Wishadell.
Okay, I have you.
My question was for the introduction to biology worksheet. I just noticed that on the questions, there's like page numbers and parentheses of in the book. And then in my book, the content doesn't match that. So like an example is like the second question, it says it's in the text page 6 through 8. And in my book, 6 through 8 is the preface of the biology book.
Well, are you able to find the relevant section by looking around?
Yeah, I did find some stuff. I just wanted to let you know that I don't think that the pages are right.
Yeah, in the syllabus, I had made a note indicating that there are several versions of this book. And depending on which version you have determines the page numbers. So it's not surprising that you would find the page numbers are offset from what the worksheet says. But hopefully you and everyone else who has this problem can end up finding the right place just by looking around close by.
Okay, thank you.
Anything else?
Hello, Professor.
Yes,
I have a question.
your name?
My name is Anna Mendez.
Okay, got you.
Perfect. So my question is for the quizzes. I saw that we only get one attempt. Is that for all of the semester? Is that going to change or is that going to just stay just like that?
Probably stay that way unless I have some reason to change it. What I'd like to make sure is that no one is constrained by time. In other words, I want to measure what you know, not by how fast you are. So there will usually, if not always, be a time constraint, but I would like to have an adjustment so that you can do the questions on any given quiz or test in the amount of time that I allow.
Okay, thank you.
Okay. Anything else?
Professor,
Yeah, your name,
Rosemary,
your last name?
Krolik, Krolik.
Yeah, okay, I've got you, thank you.
As for the lectures, will you be going over like a chapter a day or a chapter a week? Just so I know for the reading.
No, yeah. The readings I
That
will
makes sense.
assign. And the reason for that is that the lectures don't follow the textbook.
Okay.
They're wildly different. So when there's something in the text that's relevant to the topic, I will assign it. For some topics, there won't be a textbook section for you to read.
Okay, I think I missed the first part of that, of what you said, because you cut out. I'm so sorry.
I said that the lectures, the sequence of lectures do not follow the textbook.
Okay.
But whenever there is something that I want you to read in the textbook, I'll assign that. I'll post it so you'll know what to read.
Okay.
Hi, Leanne Metzger.
Okay.
Yeah, I've noticed, I know you say some things aren't graded, but in Canvas, when it says like assignments or grades, it'll have like 0 or missing. You'll adjust that as we go, right? Because like as of right now, it's telling me I have missing assignments or like 0 points out of whatever, and that'll just adjust as you do non-graded assignments, correct?
Yes. For all the assignments so far have been ungraded. So in the grade book, there are zero points available and 0 points assigned to you. But that's going to change soon, starting next week.
Professor, I have a question.
Name?
Kiyomi Anderson.
Okay.
Would you prefer for the worksheets, the ones that we do turn in, would you prefer for them to be typed out, or is it okay if they have our handwriting on them?
Either way, if your handwriting is legible, that's fine.
Okay. Thank you.
Okay. Anything else?
Yeah, I had a question. My name's Abigail Beck.
Abigail Beck?
Yes.
Okay.
A little bit later
Yeah,
question.
I got you.
apologize if it's a question that was already asked, but for the do you want to study or learn, I was confused about what you like actually wanted us to submit since there was no questions on that.
And what assignment are you referring to?
The to study or learn.
To study or learn? That's for your benefit.
Okay.
It's hopefully give you some tips about how to make your study time more efficient. So there's nothing to turn in there. Read that, derive any benefit that you can from it, and then go from there.
Okay, I see. Thank you.
I have a question about the quiz.
Okay.
My name's Katrina Bibbard.
Okay.
So for those who haven't taken the quiz yet, do you want us to email you if we don't have enough time, or tell you next week during Monday class?
Yeah, you don't need to notify me if you don't have enough time. But Well, maybe it would be a good idea. Yes, I need to know how many of you don't have enough time for this sample quiz. So if it's common throughout the class, then that's a problem I have to fix. Either make the quiz shorter or the time longer. So yeah, let me know.
Okay.
Who else? I'm pleased that you're speaking up. It relaxes the atmosphere.
Hi, my name is Abdurrahman Al-Hamawandi.
Okay.
I have a question. So for the quizzes, is there like something we study off for the quiz, or is it just about what we discussed in the class?
Yeah, what we discussed in the class. So
Okay.
to reiterate, the PowerPoint slides are very important, and they will always be in the Canvas container. I will try to get them in. Usually I tinker with slide sets right up until class time. So I usually don't deposit them until after class, but I know some of you like to print out the slides before class so that you can take notes on them. And so for that purpose, I may deposit a slide set before class, and then if I change it in the meantime, I'll replace it later.
Okay.
But the other thing to remember is, and to remember, especially when you're studying, is the recordings. So each of the sessions, like right now, I'm recording this, and I send you the URL for each recording. I post it so that you can reconstruct every lecture and go over the PowerPoints and then review worksheets, review previous quizzes and tests, and then any notes that you take and any textbook assignments. You should go over that before a quiz or a test. And I'll notify you between now and Monday about when the next quiz will be. We'll have you, in most weeks, we will have either a quiz or a test.
Gotcha.
Professor, I have a question for the quiz.
Okay, your name?
Yun, Y-U-N.
Y-U-N, okay. Okay, what's your question?
So for the quiz, do you mean by when, for example, if we study the chapter 2 and then we have a quiz about the chapter, exactly everything that study in the chapter 2, right?
Yes, if that were the case, I don't think we'd ever have a quiz that would be restricted to a particular part of the textbook. Any exam will include whatever is covered in lectures in addition to anything assigned in the text.
Okay, thank you.
All right, I'm going to share the screen now. You should see the methods of scientific inquiry. Do you see that?
Yes.
Fills your whole screen?
Yes.
Okay, that's
Yes.
what this slide set is about. That's what we're going to consider today. And if we have time, at the end, we'll go over the worksheet that most of you have done by now. How do scientists investigate the natural world? What methods do scientists use? Well, a lot of different methods, and we'll go over some of those in this lecture. To investigate natural phenomenon, you need to apply logic, analytical skills, and above all, perseverance. For all the successes you have, you'll have many more failures if you're an investigator. And scientists get frustrated just like everybody else. Now, this is a flow diagram of the so-called scientific method. You're probably all either familiar with it or you've heard about the scientific method, and it's often portrayed like you see it here in a straight line, step-by-step process, starting with you make an observation. And based on that observation, you make a hypothesis to try to explain what you saw. And then you do experiments to either validate or invalidate your hypothesis. And if your experiments explain the hypothesis entirely, and you can do it over and over, and other people can repeat your work, you become convinced your hypothesis is correct, and it becomes a theory. And we'll talk about the difference between hypothesis and theory later. However, if you do your first experiment and don't get the results that support the hypothesis, then something's wrong. Either the hypothesis is wrong or the experiments are wrong. So you adjust both until finally, by process of trial and error, you get to the point that experimental results do support the hypothesis. Now, this is what you'll see in textbooks often. It's almost never that science proceeds in this orderly, stepwise fashion. You can enter into these steps at any point. You could start with a theory and go backward. You can start with a hypothesis. You can start with experiments, or you might start with none of these. You might just be curious about something. So I've put off on the right-hand side here, you can just idly muse. I wonder what would happen if, for example, I mixed red paint with green paint, or I placed vitamin pills on my plant, or poured sand and gearbox on my rototiller. You can ask yourself whimsical questions like that and can proceed into a scientific investigation based on any kind of question that you'd like to ask. So this is more trying to illustrate, give you a more realistic view of how scientific investigation is really done. You have these boxes, making observations, asking questions, hypotheses, communicating, thinking critically, all of these things. And you'll notice they're all connected by arrows, and the arrows are double-headed. That is, that you can go either way from one box to another. So this is more like a more realistic depiction of scientific procedure than the straight line set of steps. Any question about this? Okay. Is there a single scientific method? Well, you know by now, I've said that there's not a single one. The elements of scientific inquiry, we've already gone over. Plus, I'd like to add the one in orange. Testing reproducibility. So you make an observation, you do some experiments, and you get a result. The first thing you ask yourself, if you get a result, can I do this again? So you've got this result based on a procedure, a protocol that you followed. Can you go back in your lab, do that same protocol again, and get the same result? And surprisingly, very often you don't. Something has gone wrong. Either you made a mistake the first time or you made a mistake the second, but you have to refine your protocol in order to get reproducible results. Once you do, you may become convinced that your result is accurate, that it's real. So you then communicate this result to other scientists. We'll talk about how you do that later. But the real proof of the pudding is if someone in another lab can take your protocol at their location, repeat it exactly, and get exactly your results. If that happens, now you start having real confidence that your hypothesis is valid. So reproducibility is a very important part of scientific inquiry. Now this is sort of a simplistic example, everyday example of how observation and hypothesis testing works. And we've all done this or something like this. In this case, you get up in the morning and turn the switch and the light is out. Well, immediately you have to make a hypothesis or a series of hypotheses. One, the bulb could be out, the power might be out, or maybe it's just the breaker tripped. So you test each of these hypotheses by either replacing the bulb, check to see if anything else is not working, and go to the breaker board and check for trip switches. In all likelihood, one of these tests will validate the hypothesis, and the result will be yes, and you'll figure out why the light is out. So this is simplistic, but the point is most of us use the scientific method or some part of it on almost a daily basis in terms of solving problems as we go about our daily activities. All right, here's a more elaborate stepwise scientific method. The point I want to make here is that there is a scientific method, but there are other methods for other disciplines. So for example, the legal profession has a legal method, series of steps that they use. Engineers have their method, and a scheme of that is illustrated here. I'd just like to point out that there are similarities and differences. In the case of science, you ask a question, engineers define a problem. Both cases, you do background research, you construct a hypothesis, but an engineer would not make a hypothesis, but rather specify requirements. You test your hypothesis with an experiment, or if you're an engineer, you brainstorm, you evaluate, you choose the solution, and then follow up to see if that solution actually works for the client that you're representing. And on down, the final step in either case is once you have validated what works, you communicate the results to other people in your profession. So anyone have a question or a comment about this? Okay. Making observations and asking questions. Questions arise as a result of observations, as we've said. And a question can be answered in one of two ways. Either observationally or experimentally. So there are really two kinds of science, fundamental types of science, either conclusions based on observations alone or conclusions based on experiments. So what is a hypothesis? It's a tentative explanation or answer to a question. In experimental science, investigations are based on controlled hypothesis testing. What is a control? Control is a constant. Throughout the experiment, it does not change. So for example from agriculture, supposing you wanted to evaluate the effect of fertilizer. you'd have a treatment and a control. The control would be no fertilizer. The treatment would be plants with fertilizer. Another example might be from medicine, effect of a drug. Anyone imagine how in the medical field you do a clinical trial of a drug?
I have an idea.
What's your name?
Katrina Vibbard.
Okay.
So you'd have your test subjects, and then you'd give some of them the drug, and then some of them a I can't say it correct.
Placebo.
Yeah, placebo, which is basically nothing. Well, it's something, but it doesn't affect anything. And then you see the result after they take each pill.
Uh-huh. Yeah, that's on the right track. Anybody have anything to add to Katrina's explanation?
Ikari Lopez.
What's your name? Ikari
Ikari Lopez.
Okay.
Maybe having like the same amount of test subjects as in a control area as in another control area that's not controlled.
Are you talking about location?
Well, wouldn't you have to have the same amount of test subjects to even come up with a hypothesis that doesn't have that much air in it?
Yeah, you're talking about sample size. So you'd like to have cohorts of equal sample size. So for example, testing the coronavirus vaccine, the minimum sample size was about 30,000 people in each cohort. There's one other factor that a lot of clinical trials have. Can anybody think of that?
My name is Kaylin Hilberg, and I think that making sure everyone has like the same, like health-wise, like no one has really big underlying conditions.
Uh-huh. Yeah, that would be very important if it's possible. I mean, it's really hard when you're working with humans. It's really hard to get a cohort that everybody gets 8 hours of sleep a night, everybody has the same diet, and on and on. But yeah, you would like to normalize that as much as possible.
I've got a guest, Professor. My name's Patrick Dowell, Dowell.
Okay.
Should the sample be random? Or am I thinking statistics?
Yeah, the sample should be random unless there's some reason to skew it. Yeah, you'd like it to represent the population. So yeah, that's a good point.
Professor, I have a question. So for the experimental science, do they always need to have a control group to be able to see the differences between the experimental group and the control group?
Good question. Who has an answer for that?
Hi, my name is Ron Vasatsky, and I actually work in the pharmaceutical field as a manufacturing. And a lot of times, the key factor for us is the consistency in following the procedures that are listed by the scientists when we create, like when we take a molecule and make it into a drug.
So you're saying the formulation?
Yeah, it starts from the formulation and goes all the way down to using the right, I don't know, like pipette to dispense something or some. It's really super
Yeah,
consistent. Yeah.
Very stringent protocols for producing the drug to begin with. Is that your point?
Yes, that's what I was trying to say.
Yeah, yeah, that is a very good point. The drug has to be of the same quality for all the cohorts that you treat. Okay, there's one other thing that that some trials have, and that is you test the effect of the placebo. So you have a cohort that doesn't receive anything, and another cohort that gets a placebo, and another cohort that gets a drug. And it may be much more complicated than that, but that's sort of a simplified version of a clinical trial. Okay, any other thoughts anyone has?
Yes, I have a question.
Yeah.
What is discovery science the same as observational science?
Let's see. Observational. Okay. Is observational the same as what?
As discovery science?
Yes. Yeah, they're interchangeable terms.
Thank you.
Speaking of observational science, a couple of examples here. Geology, for example. Now, a geologist can do experiments, but more often than not, a geologist will go out in the real world and look at rock formations and draw conclusions from comparing one rock formation to another. So he's not manipulating them, but the observations are valid. Another example, in the social sciences are inherently difficult to investigate experimentally because you're almost always working with humans, and humans are very difficult to control and to compare. So what do political scientists, for example, what do? Well, I happen to know what one of them does because it's my son and he is a political scientist. And what he does is goes by historical precedent. And based on what has happened in the past, he will build mathematical models and run them on his computer. And his models then, if he has them constructed properly, can predict what might happen in the future based on what's happened in the past. So that's observational. There's no experimental manipulation there. He just observes the past and tries to extrapolate from that. So both observational science and experimental science are valid. They're just different ways, depending on what your constraints are, different ways at getting at the truth, at valid answers. Anybody know what this is? Speaking of experiments.
That would be grass.
Yes, it's a type of grass. Is that Patrick?
Yes, sir.
Wheat seedlings. And what do you see there? Someone else. What is?
Some probably is planted on sand and some's on a soil of some type.
That's one possibility.
Yep, the left side definitely looks healthier.
Left side is healthier, right? Is that Michael?
Yeah.
Okay.
Justin was the first one. Justin Hansen.
Hansen. Okay, got you. Yeah, the left side is healthier than the right side. And it's possible that the difference is the soil type. What's another possibility?
Maybe this is Susanna Taylor, maybe some fertilizer on one side and not the other.
In fact, that is correct. This picture could represent any number of different things, but in fact, it is a test of fertilizer. So the plants on the left got fertilized and those on the right didn't. That is a good example of a treatment and a control. Which of these is the control?
The one without the fertilizer would be the control.
That's right. Is that Justin?
Yeah.
Okay. Yeah, and the treatment is the one with fertilizer. That's the one on the left. So having looked at that, let's consider just what is an experiment. The simplest experiment that you could possibly do is this, where you have one of each, one treatment, fertilizer, one control, without. Most experiments are much more complicated than that, but the important thing, what you always try to do in an experiment is have just one variable and everything else is constant. So among variables, there are independent variables and dependent variables. So if you, an independent variable is the one that you, the experimenter, controls. You change some part of the experiment, some factor, and then you see what else changes. And what else changes is the response, and that's the dependent variable. If you look in the upper right, it compares an independent and a dependent variable. And in this case, we're again talking about fertilizer. So the independent variable is fertilizer. The dependent variable is plant response. Either measure plant growth, number of leaves on the plant, number of fruits it makes. You could use any number of parameters to see how the plant is responding to the fertilizer. Now, how do you represent an independent versus a dependent variable? Well, in the lower right, there are these two axes, x-axis and y-axis. And typically, the x-axis would have the independent variable, the one that you control. The y-axis would have the dependent variable. So in this case, where we're talking about fertilizer and plant growth, the x-axis would have varying amounts of fertilizer from 0 to some amount. And plant growth would be reflected as either little or none up to a lot. Okay, so you have the plot. Then let's consider the orange line. Someone tell me, I have my cursor down here where the line begins in the lower left. What is that representing? Don't make it too complicated. But where the x-axis is 0 and the y-axis is 0, what does that mean?
That point would be 0 because there's 0 fertilizer, or there's, yeah, right? Because it would be there's 0 fertilizer, therefore there's no plant.
No, well, there's no plant response, but 0 fertilizer.
Oh, no plant response.
Right, so that's down here. Now, as you move.
Sorry.
Yeah, go ahead.
Doris Garcia, that's the origin. So basically, the point where the experiment would basically like begin, the starting point.
That's right. Very often the axes, both axes start with 0. So if we have 0 fertilizer, we'll have 0 plant response. Okay. Now, if we move to the right and the curve goes up with respect to the y-axis, moves to the right on the x-axis, what's happening?
The plant is growing.
That's growing.
Yes, growing.
It's responding, the dependent variable is responding to the independent variable.
Right. That's correct. As fertilizer increases, the plant grows faster and faster. We keep going up the curve and finally we come to the top. What's happening here?
Start to decreasing.
It stopped.
the amount of fertilizer has gone up, but the plant has not grown any faster. And so if you move then farther to the right, the curve goes down. What's happening there?
Fertilizer's killing the plants.
Yeah. Succinctly?
I mean, that basically too much fertilizer, I guess.
Too much of a good thing, right?
Yeah.
So, but seriously, the point is that you need to be able to plot your results and interpret it. And I emphasize this because I have found that very often students, when they're first starting to study biology, don't really appreciate the importance of a graph, or how to draw a graph, or how to read a graph. So that's going to come up again in the course. We'll have examples of graphs on tests, and so you need to know all the different parts of a graph and how to interpret. Okay, let's move on. Okay, here's some questions. A different person answer each of these. First one, scientist goes to Alaska 3 years in a row, counts the number of polar bears in a particular area. Is that observational or experimental?
Kiyomi Anderson. That would be observational.
Okay. Anybody disagree? Kiyomi's right. All right, let's go to the next one then. A scientist determines the DNA sequence of a gene that's responsible for cystic fibrosis. Experimental or observational?
Patrick
Hi.
Dowell here.
You go ahead. Sorry.
Patrick Dowell here. That would be experimental because he's gonna have to do some stuff.
Anybody else have an idea about that one?
Rosemary Krolik.
Okay.
Probably would be observational because he is observing the DNA sequence in probably multiple controls.
Okay, anyone else have a thought about it? Think about a DNA sequence. What is it?
I changed my mind. I think it would be observational because he's just going to be looking at the DNA and observing it and then determining things off of it. He's probably not going to be altering or doing things to the DNA. So he's probably observational.
Okay, I would agree with that. It is observational because a sequence is just that. It's a sequence, and there's no description here of any manipulation. There's no treatment, no control. Just had a batch of DNA, ran it through the sequencer, got some A's and T's and G's and C's. That's his observation. Now, something can be done with that. Information can be derived from it. Experiments can be done based on it, but the sequence itself is just an observation. Scientist determines the rate of growth of plants given different doses of a new fertilizer. Okay, we've gone over this already. What is it, observational or experimental?
Experimental.
Someone said experimental?
Yeah, Katrina Vibert.
Yeah, it's something we've already talked about previously. Scientists take a submarine to the ocean floor and look at the organisms there. What's that?
Hi, Aiden Dittmar. Okay.
Go ahead.
Susanna answered. She
Oh,
said
no,
observational.
Go ahead.
Yeah, it's observational.
Yeah, that's pretty straightforward. All they're doing is just looking. Scientists test the growth rate of a newly discovered bacterium on different types of food source.
Experimental.
And why?
Because they're using different food sources.
Yeah, so what's the control and what is the treatment here?
The control would be the bacterium, and then the the food sources, right? Or no, sorry, food sources would be the control.
Food sources are the independent variable. They're what you are controlling.
Oh.
And what's the dependent variable?
The growth rate.
Right. Right, it's the growth rate, it's the response. So this is an experiment, a fairly straightforward one where you have a treatment and a control. And you have an independent variable, the food sources, and you have the dependent variable, the growth rate of the bacteria. Okay, good. I'm going to hurry along here a little bit because I'd like to finish this. I must tell you that whenever I give you a test or a quiz and take time out of your day to do that, I shorten the lecture by that amount so that I'm not taking up more of your time than the program calls for. Anyway, since you're taking 10 minutes for the quiz today, the lecture will not be more than 65 minutes. Okay, proposing a hypothesis. A hypothesis is a possible answer for what you've observed. It's different from a theory. A theory is a broad set of principles based on that makes predictions, and that's a critical part of a theory, is that it's reliable enough that you can actually predict the future from it. Now, multiple hypotheses should be proposed whenever possible. It's critical that whatever hypothesis you come up with, it must be testable. You must be able to do a manipulation, and hopefully an experimental manipulation, to see if it's true. And it must be falsifiable. It must be possible, if it's not true, it must be possible to disprove it and prove that it's not true. So in biology, any proposition can be proven false. It cannot be proven true. Well, that's true in biology and in many other fields, not in others. So for example, in math, a theorem can be proven to be true. So when we say that something in biology cannot be proven to be true, that's the case for biology. So hypotheses can be eliminated, but can never be confirmed with absolute certainty. There's no absolute truth. Now, in the environment, and I suppose it's not only true for our contemporary environment today, but it's always been true, that we have science and we have pseudoscience. If you check in with social media, you'll see all kinds of things being claimed and all kinds of things being believed. So this little chart just contrasts science and pseudoscience. Changes with the evidence is scientific. Fixed ideas that don't change are pseudoscience. Science considers all the data. Pseudoscience focuses only on the data that support the conclusion. Science starts with an observation and ends with a conclusion. Pseudoscience is the opposite. It starts with a conclusion and then looks for evidence to support it. Science gives repeatable results. Pseudoscience doesn't have to be. Science invites criticism. In fact, science thrives on criticism, showing the flaws in someone else's thinking. Pseudoscience resists that. And science is driven by the desire to understand. Pseudoscience is driven by cultural or commercial goals or ideologies. So we're all exposed to these juxtapositions on a daily basis, and it's good to keep in mind whenever you see something, ask yourself, is this based on science or is it pseudoscience? How is a hypothesis different from a theory? On your own time, and this slide set will be in your Canvas container, think about these differences carefully because they will come up again. Definition of a hypothesis, it's a suggested explanation, whereas a theory is well substantiated. It's more than just a suggestion. Hypothesis based on a possibility, a projection, or a prediction, but the result is uncertain. Whereas a theory has been verified. It's been tested repeatedly, and there's a scientific consensus that it's true. Both are testable, both are falsifiable. A hypothesis is not well substantiated. A theory must be. The data are usually very limited for a hypothesis, whereas the extensive data to support a theory. The hypothesis is very limited observations, whereas the theory is based on a general principle that's been established through many, many tests and experiments. And finally, a hypothesis presents an uncertain possibility that can be explored further, whereas a theory explains a large set of observations that are consistent. Different people make the same observation over and over again. If you drop a ball, it always falls to the earth. No one has ever dropped a ball that didn't fall to the earth. Okay, but the final point about this is that there is no sharp line distinction between a hypothesis and a theory. One merges into the other. So a hypothesis remains a hypothesis as long as it's not well substantiated. But once it is generally accepted by the scientific community, then it can be called a theory. Now, one other point to be made, the uses of the term theory. Theory in everyday conversation as opposed to theory in science. In day-to-day speech, theory often means a guess or some unsubstantiated idea. For example, I have a theory that gremlins are hiding my keys. It's not a theory at all. It's a guess, it's a crazy idea. But the term theory is often used in, say, the news media, when they're really meaning a hypothesis or a guess. So be aware of that when you're reading the news or listening to a broadcast, that in everyday use, theory is used loosely, not precisely, whereas in science, it is a completely different concept.
Professor, can you give an example of a theories?
Example of a theory. I just used the theory of gravity. It's actually gravity is so well established that it's considered a law. It never changes. A theory, the cell theory. We'll come to that later, but does anybody know what the cell theory is? I'll tell you one part of it. Cell theory starts out by saying that all living things are composed of cells. And why is that a theory? It's well substantiated. No one has ever found a living thing that is not a cell. Does that answer your question? You wanted any?
Yes, kind of. Thank you.
Okay, I've got 5 more minutes and I have to let you go. Just want to make a point about graphs and thinking critically. So when you do experiments or make observations, you gather numbers, data. And usually those numbers are compiled initially in a table, for example, in an Excel spreadsheet. So you see a table here on the left. If you look at that, does anything jump out at you? You see any pattern? I think not, at least I can't. But if you take numbers like that and you plot them on an XY graph, almost always you'll see information immediately jump out at you. So on the right is a wildlife populations, and you have numbers of animals plotted against time. If you look at bears, the blue line, you can see that over 5 years, the population of bears is continuously increasing. You see that immediately. If you look at dolphins, you see the opposite, that over 5 years, the population has gone to 0. Whereas with whales, population hasn't changed much over 5 years. So my point is that when you graph something, when you display your data in some type of graph, you get a lot more information from it than if you just look at a set of numbers. Okay, there are numbers of ways. Once you get a result that you've worked very hard to get, you've confirmed your hypothesis over and over in your lab, you want other scientists or the whole world to know about it. And so how do you communicate? Well, most science is published in scientific journals, like the journal Science or the journal Nature. There's another series of journals called the Public Library of Science, where a lot of good science is being published these days. And to get published in one of these so-called peer-reviewed journals, you submit your manuscript and it is sent to your peers, 2 or more of your peers, other scientists working in your field, very familiar with your field. They will critique it and they'll decide if it's publishable, if it isn't publishable, or if it would be publishable if you made some modifications. And so if you do, very often it'll appear in print. So that's one way. Most scientists attend scientific meetings frequently. They actually meet their peers face to face, if it's not a pandemic. And a lot of scientific communication goes on between scientists at meetings. And there are other ways. There are seminars. There are special events where scientists exchange information. The usual dogma is that if you're in a field, or you think you're in a field, and you don't know the data before they're published, then you're not really in the field, or you're not communicating very effectively. Now, I am actually over time. This last slide, I call it thought problems. What I'd like you to do is on your own time as a homework assignment, go over these and answer the questions as best you can. And these types of questions or similar are likely to come up on subsequent quizzes or tests. So I'll let you go. I'm going to stop sharing. Anybody have questions or comments before we end the session?
Thank you, Professor.
Okay. All right, well, thank you all. Thank you for contributing to the discussion. I appreciate that.
Thank you, Professor.
Okay, I'm looking forward to seeing you on
Thank you,
Monday.
Professor.
Okay,
Thank you.
all right. Bye.
Thank you, Professor.
Thank you. Bye.
Take care.
Thank you.
Have a good day.
All right, I'll do my best. You too.
Professor?
Yeah?
Hi, I'm Kenya Vidarrazaga. I wasn't here for the first class. I just want to let you know, if just in case you took attendance.
Yeah. Okay, well, thank you for letting me know.
Thank you. Bye-bye.
You know about the recordings, right? You have a URL so you can listen to that class all over again.
Yes, I did.
Okay, good.
Thank you.
All right,
Bye-bye.
bye.
Professor,
Yeah?
I have a question, please.
Uh-huh.
You teaching just bio 130,
Yes.
not 131?
No, 131, I think, is the lab.
It's lab, yeah.
Yeah, no, I don't teach the lab. I have 2 sections of 130, and I'm not involved in the lab at all.
Okay. Yeah. Okay, thank you so much.
Bye.
Thank you. Good evening. Bye-bye.